Thank you very much. Today I will talk on nano-size compressed product in vitro release testing. I'm a research chemist from Office of Regulatory Affairs. So I will start my presentation with introduction to drug product containing nanomaterials, liposome and liposome drug product, in vitro release testing of compressed product containing nanomaterials and challenges. Finally, I will discuss innovative method we developed in our lab. So what is complex product? Drugs that are characterized by complex active ingredient, formulations, or route of delivery. What is drug product containing nanomaterials? Submicron size particles with one or more therapeutic agents that are dispersed, adsorbed, or covalently bound in encapsulated vesicles, capsules, or polymer matrices. Drug-loaded liposomes, polymer nanoparticles, or protein nanoparticles are prime examples for those products. So what is liposome? Liposome is microvesicle composed of lipid bilayer. It has a central aqueous cavity. We can use liposomes to deliver drugs. If active pharmaceutical ingredient is hydrophilic and water soluble, we can load into the central cavity, aqueous cavity. If active pharmaceutical ingredient is hydrophilic, we can load into the lipid membrane. There are many different kinds of liposomes based on the structure of lipid membrane arrangement. They can have a single layer of single lipid bilayer is called unilamellar liposomes. Multiple concentric lipid bilayer is called multilamellar liposomes. And they can have a multiple compartment in single liposomes separated by lipid membrane. It's called multivesicular liposome. When liposome is loaded with active pharmaceutical ingredient, it is called liposome drug product. So how we do in vitro release testing for liposome drug product? So we set the condition for drug release in drug release media. We can control pH, temperature, and media composition and other parameters. Then we add the liposome formulation to the media. So it starts releasing active pharmaceutical ingredient and to the outside of the liposome. Then we collect the sample each time point. Once we collect the sample, we need to separate them immediately. Collecting sample does not mean it stop releasing. Even we taking the sample out, it keep releasing. So we need to separate them immediately and then quantitate the released drug to make a drug release profile as shown in last diagram. But this is challenging process because both nanoparticles and active pharmaceutical ingredients are same size. So it is difficult to separate by conventional technique. And also these liposomes are delicate structure. If you use harsh conditions to separate them, they can break open. So when we develop a drug release testing method, we need to consider all these parameters. There are mainly two different ways to do drug release testing for liposomal formulation. One is as shown in previous slide. We can take sample and then separate externally, either filtration or centrifugation. But both filtration and centrifugation is challenging because active pharmaceutical ingredient can bind to the membrane or liposome can get disintegrated. Sometimes difficult to centrifuge because lipids and media has same density, so they don't get sedimented with centrifugation quick enough. Other method is membrane diffusion method. We can take a dialysis sac and then load liposome inside the dialysis sac and place dialysis sac in the drug release medium. And drug release out from the liposome, release to the surrounding medium, and then we can take samples from surrounding medium. We can do in the container, or we can do this on flow-through system like a USB type 4 dissolution system. But this is also challenging because active pharmaceutical ingredient can bind to the membrane in this case. Also, it's a non-sync condition most of the time. That drug release inside the dialysis cassette that does not come out immediately. There's always concentration gradient. with considering all the above challenges, we set up the condition to develop new drug release testing method. We need to eliminate manual separation, avoid artifact associated with disruption of nanoparticles, and we need to do it automated way and real-time sampling and analysis, because even after we take the sample, active pharmaceutical ingredient keep releasing, we cannot store the samples. So first example I'm showing is application of capillary electrophoresis to do automated drug release testing. Capillary electrophoresis is a very gentle separation technique. It uses buffer-filled glass capillary, and once we inject sample from one side, we can apply electric field and we can separate released drug from liposome-bound drug. And then we can use optical detection, like the optical absorbance or fluorescence. Even we can connect to the other detectors and then measure the released drug. So this is automated method. And this buffer we use in this method for separation is compatible with liposomes. And this method need very small volume of sample. It is a fraction of 1 microliter, few hundred picoliters. And also we can do simultaneously take the sample out, separate and quantitate in automated manner. So this is the example of use of capillary electrophoresis for drug release profiling of liposomal doxorubicin. In this case, the doxorubicin is loaded inside the liposome. Once we set the drug release condition, it starts releasing active pharmaceutical ingredient. And then we can set up the instrument to take the sample directly from the drug release vial and then separate from bound drug to pre-drug as two separate peak. And then we can integrate the peak at the end of the experiment and make a drug release profile. Here I am showing a drug release profile of 5 FDA-approved doxorubicin liposomal formulations, and more details available in a recent publication. We can use same method for drug release profiling of VISUDYNE. VISUDYNE is a liposomal formulation of verteporfin. Verteporfin is water-insoluble compound. So once we inject this formulation to our blood circulation, it releases as a human serum albumin complex, as it is water-insoluble. In this case, we use human serum albumin to assist the active pharmaceutical release as a complex, and we can separate the released drug from liposomal bound drug. So drug release condition for this drug is we need to get 90% of active pharmaceutical ingredient released in 10 minutes. And we changed the verteporfin to human serum albumin ratio and did the experimentation. And even at 1:1 ratio, we can get out 90% of the active pharmaceutical ingredient. In the human blood, we have about 35 gram to 50 gram of human serum albumin, and it has much more human serum albumin than it wanted to release the drug. So this experiment was very successful, and more detail available in recent publication. So next, I'm going to show a different way of doing drug release testing. In this case, we use electroanalysis. So we insert 3 electrode to drug release container or drug release vial, and then we can in situ measure the released drug without taking any sample. So here in this case, we use the redox reaction of doxorubicin, whatever coming outside the liposome, and then redox current is proportional to the concentration of doxorubicin. So in that way, we can measure the drug release in situ without taking any sample, and then we can generate the drug release profile as shown in this figure, and more detail available in our recent publication. We also develop other method. One of them is a chromatographic method. We modify HPLC system to develop a drug release profiling method. Here we use a monolithic silica column coated with polyvinyl pyrrolidine. And this column has 1 micron through pore, so those pores go all the way along the column. And when you inject the liposome to this column, it let liposome pass through the pores and then release active pharmaceutical ingredient, bind to polyvinyl pyrrolidine, and then we can elute them later. So we can separate liposome from released drugs and generate the drug release profile. This is the drug release profile of FDA-approved 5 liposomal formulations. And again, more information available in recent publication. So each of these methods has advantages and limitations. So we have to carefully select the method when we use it. Capillary electrophoresis use very small sample volume, so that's an advantage. And also it's a delicate separation technique because separation happens in aqueous phase. But separation is done by applying electric field, and only charge active pharmaceutical ingredient can be separated by this method. That's the limitation. Electroanalysis is another way to do the in vitro drug release testing. So this is advantage, no separation, no sampling. We can set up the instrument and it measure the released drug automated way. But this method only work with electroactive APIs. So these compounds should have a redox or oxidation reaction. The other method I discussed is a chromatography-based method. This method highly accurate. It take exact sample volume. If they take 1 microliter, it's exactly take 1 microliter, and it analyze the sample. Also, we can use the multiple detectors. Some of the active pharmaceutical ingredients are not optical detection cannot be used with some of the APIs. So we can even set up to the mass specs for quantitation. But this method need a specialized column compatible with the formulation. If we use a column which is not compatible, that can break open the liposome and then give erroneous results. Finally, I would like to summarize my presentation. So we developed an innovative method for drug release profiling with a real-time sampling and analysis for liposomal product. Also, active pharmaceutical ingredient specific physical and chemical properties should be considered for selecting in vitro release testing. So we have to look at as I shown in these 2 points, we have to look at the compatibility of the liposome formulation with the method. And also detection method has to be compatible with the active pharmaceutical ingredient. And these methods can be utilized for other liposomal formulation with careful selecting the compatible method. Finally, I would like to acknowledge my co-workers at Arkansas Laboratory. They are the one who did all these experiments, including my lab director listed at the end. Also, I would like to thank ORS, OGD, CEDAR. This work done as a collaboration between myself and Dr. Wenley. And also I received technical support and financial support from CEDAR, OGD, ORS. Also, I would like to thank Office of Regulatory Affairs, Office of Regulatory Science. They provide financial support and technical advice for my work. Thank you for listening my presentation. Next speaker is Dr. Dong Kai Shu. I'm inviting him to give his presentation.